Forensic Science Lecture 10-4, Mitochondrial DNA and Collecting Biological Evidence. Human cells contain two types of DNA, nuclear and mitochondrial DNA. We know that the nuclear DNA is found in the nucleus, consists of 23 pairs of chromosomes, inherited from both parents, and these are linear chromosomes. Mitochondrial DNA, on the other hand, is found outside of the nucleus, and it's only inherited from the mother and consistent or is in loops instead of linear DNA. Remember, mitochondria are the organelles responsible for 90% of our energy body needs. They contain several loops of their own mitochondrial DNA, all of which are involved with energy generation. Each cell contains hundreds to thousands of mitochondria, depending on the type of cell it is, which results in hundreds to thousands of mitochondrial DNA in each cell. Forensic science rely on mitochondrial DNA when nuclear DNA is significantly degraded, such within charred remains, or is not present at all, or is present in too small quantities. Because mitochondrial DNA only comes from the mother, all individuals of the same maternal lineage are indistinguishable by their mitochondrial DNA. So they can be linked to a family, but not necessarily individualized. Mitochondrial DNA analysis doesn't approach the sensitivity that STR analysis does. So the discriminatory power of it is much, much less. It's also more rigorous, more time consuming, and more costly than nuclear DNA profiling. So it's typically reserved for samples where STR analysis may not be possible. But each loop of mitochondrial DNA contains enough bases to make about 37 genes, but there have been two regions that have been found to be hypervariable in humans. They refer to those as HV1 and HV2. These regions are processed through the PCR process to amplify them, and then the order of the bases is determined. That process is known as sequencing. The labs then report how many times these sequences have been repeated to the FBI mitochondrial database, which permits an assessment of how common or how rare these sequences are in the population. We've already talked about CODIS, or the Combined DNA Index System. We know it's a computer software program developed by the FBI that maintains local, state, and national databases of DNA profiles from convicted offenders, unsolved crime scene evidence, and profiles of missing persons. And it's probably the most significant tool to come from DNA typing. The ability to compare DNA recovered from a crime scene to criminal offenders, unsolved crimes, missing persons is completely invaluable to investigators. And as I said, one of the most significant tools that they have. Currently, CODIS has about 470,000 profiles from unsolved cases alone. And so it helps to connect crimes in that manner and helps to come up with a suspect when there may not be one readily available. The amount of biological evidence or DNA that is required by DNA profiles is as small as 125 picograms, which is a trillionth of a gram. Human cells contain 7 picograms each, so that requires only 18 cells in the sample. With current technology as they're developing, this number can be lowered or has been lowered to 9 cells. This is known as low copy number, but because it's still in the process of development, it's not fully admissible in court. But the progress is continuing in that manner. Types of biological evidence have also been extended with the technology that now exists. to stamps and envelopes that have been licked, cups or cans that have been drank from, gum, the sweatbands of hats, bed sheets with dead skin cells on them, um, even epithelial cells, which are skin cells, can be transferred to weapons, insides of gloves, or pens that have been used by a suspect. This is known as touch DNA, which again is only going to provide a small amount of DNA evidence, but with our technology, we can amplify that, especially with the PCR process. When we talk about collecting biological evidence, we've discussed it before. That before collection begins, all evidence should be recorded through notes, sketches, photographs. If the position of the blood can provide information to investigators, an on-the-spot evaluation must be performed prior to the collection process. Um, and of course, evidence technicians must handle all body fluids and biological material with minimum amount of personal contact. So the use of latex gloves, changed often throughout the process, face masks, lab coats, eye protection, um, uh, shoe covers, and possible overalls is highly important to prevent that contamination. When packaging biological evidence, we know that plastic and airtight containers must be avoided at all costs, that the accumulation of moisture can contribute to the growth of DNA destroying bacteria and fungus. So each article should be packaged separately in paper bags or well ventilated boxes. And then, of course, a biohazard label must be on each container. If it's feasible, then the entire article should be packaged and sent to the lab. If it's not, such as dried blood on a large area, it can be removed with a sterile cotton swab moistened with distilled water and then rubbed on that stain, and then air dried, because nothing should ever be packaged wet, and then packaged according to the proper packaging procedures. Along with the sample of the stain itself, a sample of the unstained surface should also be collected and packaged for comparison. This is known as a substrate control, and often they use it to compare that it wasn't a contamination of the normal material of the item. And then reference samples from the victim and the suspects are also collected regularly in order to use as a comparison to the evidence that was collected. Most of the time, this is obtained through a buccal swab, which is a cotton swab which is placed in the subject's mouth on the inside of the cheek and vigorously swabbed. This transfers the buccal cells, or the cells from the inside of the cheek, onto the swab. But it can also be, if the individual is not available, can also be obtained through a toothbrush, combs and hairbrushes, razors, dirty laundry, used cigarette butts, and earplugs as well. When packaging biological evidence, after it has been collected and packaged correctly, it then needs to be refrigerated or stored in a cool location to not degrade any of the biological evidence. The only exception to this would be blood that is found in soil. Soil contains microbes that can rapidly degrade the DNA, and so they need to be stored in a clean glass or plastic container and immediately frozen to prevent those microbes from destroying the DNA. Contamination of DNA is always a concern for investigators. It can occur by introducing foreign DNA through coughing, sneezing onto the evidence as it's being collected, or through incorrect packaging and the cross-contamination of materials. But the STR process can actually indicate if contamination has occurred. Typically, 2 band patterns are shown with STR, one from each chromosome. If there are more than 2 bands present in the reading, then this suggests that contamination has been or has occurred. This ends lecture 10-4. There is a quiz next class.